There was a question that came in about, I've been seeing more reports of nuclear dense fine speckled pattern. My opinion is this doesn't correlate with connective tissue disease. Do you have any thoughts on this? And P.S., you never told me about this as a fellow. That came from Dr. Scott Zashin, but then you addressed it and Dr. Zashin wrote back, thank you. So anyway, a couple of questions on COVID infections and ANA. Peter, you want to start out with that? Or vaccines, COVID infection or vaccines and ANA?
So there have been a number of reports now about various autoantibodies in COVID infection. And some of these have come from Yale, from Dr. Irasaki, some from Stanford. P.J. Utz has written quite a bit about this. And a question that has come up is whether or not actually COVID is inducing this, or you're actually seeing this in people who are selected and have low levels of antibodies. That's not really been clear because we don't have very much in the way of longitudinal data. The other thing that is interesting is that the bulk of the autoantibodies that are coming up are not particularly those that are found in lupus. They are found occasionally, but it's much more anti-cytokine antibodies, anti-cellular antibodies, a whole bunch of different antibodies. So when you look across the board, it doesn't appear that particularly the autoantibodies in lupus are induced in any kind of a reproducible way by COVID infection.
Okay, we'll turn to Dr. Troy at the front microphone.
That was a great comment and great talk. That was the line of question that I was going to say, because we've definitely got of patients who were tested positive for ANA after they had COVID. And not only ANA, in fact, they tend to also have anti-GP to glycoprotein 1 antibody and lupus anticoagulant if they have lupus. What we did notice is that they tend to get this spike of titer, and the titer drop quite rapidly. And at this moment in time, all we're doing is just hanging on, waiting to see what happens, because we don't know what is happening.
You know, there was a story for that in the past with rubella in children. And, you know, there is an association of anti-phospholipid antibodies in COVID with the thrombosis of COVID, this widespread microangiopathy, which really resembles catastrophic anti-phospholipid syndrome. I think we only have one immune system. And there's also a great deal of work that was quite popular about 10 years ago or 15 years ago, pointing out that autoantibodies are actually in service to immunity. They're there for a reason, but when you're healthy, they get covered up. And if you believe in the urine network, or if you just believe they get covered up some other way, and that really pathology is when they just run rampant and out of control, but in fact they have good service to balancing of the immune system. So it makes sense that any virus, especially a bad one that's affecting, you know, a lot of people at the same time that everybody's worried about and testing things on, is going to be associated with some autoantibodies. But to the extent that they're transient, I think we go back to the rubella thing. You just do exactly what you said. You watch and wait and see if there's anything to worry about.
Okay, there's a couple of questions on anifrolumab safety. One about using anifrolumab with severe leukopenia, and one about checking annual quantiferon testing. Dr. Narain, you want to start us out, and then the other panelists can weigh in?
Yeah, I don't think that there is much data to suggest you need any kind of quantiferon testing for anifrolumab other than what we're already doing in our population. With respect to leukopenia, you know, obviously there is an increased risk of infection in this population, so we have to be wary of that, especially in this group. I don't have anything much to add beyond that.
Okay. We'll go to the front microphone here.
I have a treatment question. Is there any reason we actually have to use DMARDs with treatment of lupus? Now we have 2 new drugs. And concerning the treatment of Saphnella versus Benlysta, are we going to make decisions based on the lab work on a high interferon profile, or are we going to go back to what we used to do, which is just treat people clinically? Thank you.
I think the right answer is treating clinically, obviously, in any situation. I don't think, again, with interferon gene signatures, that data is really fraught with reliability of the interferon gene signature, depending on which platform you're using to test it, and also what do you do with that data that I pointed out? There's so much variability in terms of placebo response in those groups, and what that pattern means with respect to response to the drug. So my recommendation would be to treat clinically, see the patient in front of you, and make the decision with partnership with the patient.
I would just say that, you have patients in your practice, and if they're doing well on methotrexate or mycophenolate, there's no need to switch them. And I think these drugs are not quite as expensive. Mycophenolate's not cheap, but you could use mycophenolic acid, which is a little cheaper. But if you can, with a reasonably low cost, treat a patient, that takes care of many of your uninsured patients, and it takes care of those people whose insurance companies are very difficult to get approvals from. You usually have to come up with a good reason to get a really expensive drug.
I think, based upon what we heard from Ernest Choi this morning, where there are a dozen biologics available or more in rheumatoid arthritis, and the idea of treating blindly and waiting for a response may not be the best way to go. And as these, new drugs become available, and we'll expect there be more, having a way to provide decision support to a physician with regard to which of these drugs might be used in which order might really improve the practice of medicine and perhaps get us toward the idea where we do have some more likelihood of precision medicine. I think at the end of the day, the clinician makes the decision, but the more information the clinician has, the more likely it is that the patient will benefit and not have to go through these periods of trial and error.
And I also think that the regulatory atmosphere is working against us here, because maybe we're not scientifically knowledgeable enough or experienced enough or have enough funding to do the kinds of studies that are needed to be 100% certain which drug you should pick for an individual patient. But if we had data that suggested you could take a 40% response rate up to 70%, we should be allowed to use it. And it's going to be a really hazardous regulatory environment to get that through. So I want to ask a question of this entire group. Would you rather just have these drugs that look theoretically promising and use them empirically? That's A. Or don't you wish you had the drug and the test? B. Who wants A? Really? The drug and the test is going to tell you, at least not with 100% certainty, but with better confidence, that this patient is more likely to respond.
I mean, the point here is that I think that the new drugs that we have, Benlysta, anifrolumab, have effect sizes of basically 10 to 15%. That's pitiful. And so, however, if you had the opportunity to identify which people were likely to respond, you probably would be much more comfortable telling your patient, you have a little bit of a response and I don't have any idea what to do, so you want to take this. Or based on your profile, whatever that is, be it an interferon signature, be it something else, you have a likelihood of 50% of responding. I think that's an advance. Plus, lupus is a disease where damage is occurring all the time. You really don't have the luxury of waiting around for 6 months or a year with an ineffective therapy. And it's not as though we have such great information about when to use mycophenolate or methotrexate or any of the other dozens of drugs. We did look through the Symphony database to see what doctors actually were doing, and every drug you can imagine or they can spell has been used in some patients with lupus. So it indicates that there's a level of desperation in clinicians, and that a lot of what we're doing is grasping at straws, but being able to match drug to patient would be a huge advance in this field.
Peter, I'd like to brag about something. I have had at least 10 NIH grants rejected where I wanted to study the biomarkers of the commonly used cheaper medications.
There's also
Well, that's a world record, and congratulations.
There's also the
Jack?
practical aspect of this, right? Because a lot of our lupus patients are really young women who are in their childbearing years. So we are really limited by what we can actually give these patients to get under disease control. You know, we would all ideally like them to be in great control when they get pregnant, but the reality of it is that most of them are not, and we have to treat them through these years with some degree of safety. So as we get to know more about these drugs and study some of this retrospectively, we can make better decisions for the patients as well.
Can I ask Artie a question back about anifrolumab? Maybe somebody knows, I don't know. Does anybody have any information that actually herpes vaccination works in the presence of blocking type 1 interferon?
I don't think we have any information.
No, we don't.
Well, what you'll have is antibodies already made, but you're right, you don't know for sure. This is on purpose, you know, we don't want you to ask the question. We're scared of you.
Can you censor her? That's your role as
Where's the
moderator.
mic control, the volume control?
She's in very good behavior today.
Absolutely. We're delighted, Joan, that you're here. For years I've been telling companies that want to come up with a new drug, don't give me a new drug. Give me a drug with a test that tells me how to use it, because otherwise you're ninth on the list. And the problem is and companies actually do this research. I mean, they do invest in some biomarker discovery and whatnot, but no one has come up with anything yet. This really should be a regulatory issue. I mean, regulators shouldn't approve another drug to go on the market for a 10% advantage.
Cut his microphone off.
yeah.
Please, stop it.
Well, honestly, I mean,
Jack, he just suggested
Except in
tripling the
lupus.
time to develop a new drug.
Yeah,
That's
except
true.
in lupus
That's
where we
true.
have no
Just
drugs,
develop-
thank you.
Remember, you have to do a trial of the biomarker along with the trial of the
all right,
drug.
I'll admit
So-
that that's radical, but maybe not as radical as dispelling the ANA that you came up with. So are you telling the audience, and this is serious, what are the tests about? They're about certainty in diagnosis. They're about homogeneity in clinical trials. So there is this uncertainty about ANA. Should rheumatologists throw it out? Should they keep repeating it? Should they now rely only on the more specific antibodies of double-stranded DNA in Smith or Rowe?
Well, as I mentioned, the problem with the ANA is the heterogeneity of the test, that you don't know what you're doing. The problem which has been imposed by some insurers is that you need to do it in order to get the use of medications approved. So everything, the law of unintended consequences plays out very strikingly when, with good purposes, individuals do things that then become basically the rules.
All right, well, thank very much our panelists, and thank you the audiences, both here and virtual. It's lunchtime, and please do visit our exhibitors and sponsors who are kind enough to support us and allow us to have this meeting, and there's lunch there as well. We will come back at 1 o'clock sharp for pod number 3. So thank you very much for this morning's session.